In this case study, we focused on trypanosomiasis and its particular role within a community which was in a state of transition. So we looked into sort of health, well-being, and ecosystem change within a study area in the Luangwa Valley in Zambia. That's the team that was involved. Noreen is actually the country lead, but there's a few of, she's not here unfortunately, and Martin Simunza also couldn't come unfortunately for similar reasons, but some of our teams in the audience here, Pete and Simon and Joanna in particular. So I wanted to just introduce trypanosomes a little bit. Not all of you will be familiar with what they are, what the life cycle is. So just briefly, trypanosomes are protozoan parasites in the genus Trypanosoma. There's a very large number of trypanosome species which infect a large number of vertebrate hosts. In particular, all domestic animals are capable of being affected by trypanosomes. There's 8 pathogenic species that affect domestic livestock. And wildlife hosts are very important in terms of trips. Most mammalian wildlife species in the Luangwa Valley anyway are capable of supporting trypanosome infections to some degree. It's a vector-borne disease. The vector is an insect, the tsetse, which is a Glossina species. There's 28 different species of tsetse which occur throughout sub-Saharan Africa. In Zambia, there's only 3 species. There's cyclical transmission within the tsetse fly for most trypanosome species. Human infection is possible. In this part of Africa, there's only one trypanosome that infects humans. That's Trypanosoma brucei rhodesiense, the cause of human sleeping sickness, which is a fatal condition if not treated, and most patients die within 6 months. But infection in humans is generally spillover. Humans don't form part of the reservoir, in contrast to the animal hosts. So I'll introduce our study area very briefly. This is the Luangwa Valley in eastern Zambia. The Luangwa Valley has 4 national parks. It's a biodiverse area with a lot of potential for sort of photographic safaris, and the South Luangwa National Park in particular brings in a lot of income in terms of photographic safaris. The national parks are surrounded by game management areas which are multiple use areas, so commercial safari hunting provides a big input there, but also agricultural activities are allowed, and there's a relatively low human density compared to many parts of Africa, and this is the sort of status quo here that I'm presenting here. Historically, there hasn't been livestock keeping in any substance throughout the Luangwa Valley, most likely because of the impact of tsetse and trypanosomiasis, but conditions also not always very easy for keeping livestock. And as a result of this relatively low human density, there's been fairly consistent land use sort of patterns over the last century. Now, our study area, the place that we're interested in is this little part of the Luangwa Valley here. There's a gradient in an altitude going from the valley floor here up towards the eastern plateau. This area around Chipata, Katete, Petauke, that's a higher altitude area that's very intensely populated by humans with a lot of agriculture as well. So there's a lot of pressure on land up in that particular region. So why were we interested in studying that particular area? As I've said, there's historically been a low population density, but in this particular part of the Luangwa Valley, that seems to be changing. There's been an influx of people and new settlements into that area in the last few decades, and a corresponding increase in livestock of a variety of species. And this has resulted in sort of concurrence of land use and land cover change with potential impacts on ecosystem services. And we're particularly interested in how that's impacting on trypanosomiasis disease transmission. In general, with trypanosomes, what happens when you get modification of the habitat and land cover change, you get a reduction in tsetse populations and less transmission, but it's much more complex than that. And there's also other factors. With an increase of livestock that are being introduced, they could then become a more important reservoir for the disease. So it may switch from a sort of predominantly wildlife-based cycle to a more of a cattle-based cycle. We're also very interested in this crop here, which is cotton. There's been a lot of sort of encouragement for farmers in the Luangwa Valley to start cotton production. I'm supported by some commercial companies that provide seed and in particular provide insecticide. An insecticide is generally used as deltamethrin, which has quite profound effects on insect populations and particularly tsetse. So that can also have an impact on the disease situation. And we're also concerned that if cotton production stops, then that land becomes fallow. It could become a good habitat for tsetse. So it's unclear which way transmission would go. And another point which is important is that new interphases like this, new areas where cattle and human populations have been brought into close proximity to more undisturbed habitat, they've been identified by previous research to be particularly at risk of epidemics of disease. So in our work, we started off doing a household and animal census so that we knew the exact populations and their spatial locations within the study area. And these figures, I think, reveal that there's actually a reasonable human population in the study area and correspondingly quite a large cattle population now. Interestingly, quite a lot of pigs. Pigs are also susceptible to African swine fever, which causes quite a lot of problems in the Luangwa Valley. But that's outside the scope of this talk. The figures from Mambwe district suggest that there has been an increase in population from the 1990s through to now, but it's not been as profound as we initially sort of expected, or certainly people believed was the case. So we did a lot of participatory research and participatory methods to try and understand the key sort of drivers of human behavior and to understand where they're going, what sort of factors bring them into risk with tsetse populations in particular. So these included key informant interviews. Those were done both in the Luangwa Valley and nationally. Focus group discussions and participatory mapping sessions where we sat down and asked the communities to help develop a map to show us where the sort of most important sources of risk in terms of disease were. And then a very large structured questionnaire survey, 211 households, a very detailed questionnaire, which accompanied animal sampling and an epidemiological survey. We also had a look at the tsetse vector. There's some traditional sampling techniques for tsetse vectors, which include something called a black screen fly around and epsilon traps. What I presented here is the results of the black screen fly arounds. And what you do is you walk along a transect, normally 5 kilometers in distance, stop every 200 meters and sample the insect population of the tsetse flies that have been attracted to the screen. We did 2 surveys, one in June during the early dry season in the cold time of year, one in November when the rains normally start. But this year the rains were a bit late, so there's been very little rain. Tsetse populations sort of retract a little bit during that period. So there's a very focal area of tsetse density displayed by both of these maps. And important factors that seem to be good predictors of tsetse distribution include the distance from the national park and distance from roads and rivers. The tsetse density was much lower than we expected. Well, it was lower than is present in the South Luangwa National Park. It's a relatively low density. So we did a epidemiological survey, a large epidemiological survey designed around the census. A colleague, Joseph Mubanga, had done a PhD in the same transect in 2005, and we used that to compare to our figures. And I think it's quite striking that there's been a change in prevalence of trypanosomiasis across all livestock species. And I'll discuss that in a minute. No human infective T. brucei rhodesiense was detected. It's important to note that we use molecular methods which detects actual presence of infected organisms, not just exposure through antibodies. Migration was quite complex. Surprisingly, there was a lot more internal migration, so a lot of migration within Mambwe district rather than from outside. The hypothesis at the beginning was that people were moving from the plateau region at the top of Eastern Province down into the study area. And although that's occurring in 17% of respondents, it's less significant than we'd appreciated. But there's a general change, an increase in migration from the Eastern Plateau, which has possibly leveled off. Change has been accompanied by land cover change as well. And this is a map produced through remote sensing. Joanna Coletso has done a study of land cover change through time. And we can see here that the proportion of agricultural land has increased quite significantly in our study area, slightly less within Mambwe district, which tends to be further north. So that's closer to the valley floor. The household wellbeing questionnaires revealed some very interesting results. So there's far too much detail for me to go through in this talk, unfortunately, but significantly a large proportion of households are growing cotton, and that's bound to be having some influence on tsetse populations. Food security is poor, access to water is poor, but generally they're quite happy with their life satisfaction. So in summary, the main take-home message is really that land cover change has been significant, but particularly marked towards a plateau closer to where the new migrants from that part of the plateau have come down from. But the migration patterns are much more complex than we expected. A lot of movement is over relatively short areas, short distances. Trypanosomiasis prevalence has reduced, but importantly, the livestock population has increased, so the overall reservoir has not necessarily decreased to the same extent. And tsetse density is relatively low, so it appears that land cover modification may be reducing tsetse density, although we can't really conclude that. Importantly, another aspect that came out of the discussions with the communities, they have very little access to human health services and almost no access to veterinary health services. And that's something that I think UNZA, the University of Zambia, wants to focus on through the development of farmers groups after this project to continue that work. That's a very quick overview. I'm running out of time, unfortunately. Could I just very quickly just mention Joseph Mubanga? He was instrumental in starting our project. I did a PhD with him at Edinburgh, and it was his thesis that we used to develop this hypothesis, but he sadly passed away last year, much too young, so I just wanted to acknowledge him.